which is the minimum energy required, to start the chemical reactions.
Minimum energy required to start the chemical reaction. Okay. So when you say enzymes lowering the activation energy, what do you actually mean by that?
It means that less energy is required, or uh, there's less temperature requirement, which means that it can heat up faster and start working quickly.
Yes, we don't need to elevate the temperature of the reaction to a very large temperature without uh, having the need to elevate the temperature of the enzyme can do the same work, which is actually under normal conditions. If the enzymes were not there, it must have been provided by heat. This is the thing that is, this is important, and this concept is important. This one, which I am serving right now. So usually the activation energy is provided by heat, and it is defined as the minimum energy needed to convert a substrate into a product. अगर आपसे कोई पूछता है कि what is the activation energy? So this is the activation energy, and a body temperature of 7 degrees Celsius is not enough to give activation energy to most metabolic reactions. Degrees Celsius, but we have to have the reactions done. So for that, we provide that boost of energy by the enzymes. And looking at the enzymes now, it would have been provided by heat. Okay? And then we can sum up and we can say that in the last sentence, the enzymes lower the activation energy by holding or interacting the substrates in a specific way. और जब specific way की बात होगी, do you remember which specific way did I tell you last time? What is that specific way by means of which the enzymes can hold or interact with the substrate?
Is the sequence of amino acids at the active site?
Holding. The key word is holding. This is the enzyme, and this is the substrate. And I'm saying that the enzyme is holding the substrate in a specific manner. So what's that? By means of some bonds?
Yes, miss, the hydrogen bonds.
The hydrogen bonds, right? Okay, fine. Then we move on to the next slide, and I want you to go through this slide very carefully, the information in the slide.
Yes, miss, I've gone through it.
Okay, so what do you understand by this?
So, miss, it basically explains that the alternate metabolic pathways lowering the activation energy. That's the part it follows, and then the energy content of the substrate is not hindered at all. It's just that the course of the energy is lowered.
Course of the energy is lowered. And how is it lowered? By providing an alternate metabolic pathway.
Yes, miss.
Dekho, see, I will show you a diagram like this. One of my presentation on the enzymes. Although it's a low-level presentation, but it very well explains. Look, imagine a chemical reaction as a process of rolling a huge stone or reactant up a hill so that it rolls down and breaks into products. So you wanted to break a big stone into a product, okay? And that's your chemical reaction. Yeah. After reaction is the big stone and the product are the tiny, tiny pieces. Okay? Then imagine a chemical reaction. You want to have this chemical reaction. And can you look at that activation energy with the dotted line? बनी हुई है और उसके नीचे जो वो स्पेस बनी हुई है, that bracket, and you look at that square bracket,
Yes, miss.
right? That square bracket is the activation energy which is needed to roll the stone up the hill. And इसको ऊपर carry करना है और फिर ऊपर से हम इसको फेंकेंगे तो it will break into small pieces. So you need some energy to bring that stone up the hill. ठीक, इतनी energy आपको चाहिए. Once over the hill, the rest of the reaction occurs automatically, right? The stone rolls down and breaks into tiny pieces. Look at this. The energy needed to start a chemical reaction is called activation energy. और यहाँ पे जो उन्होंने activation energy दिखाई है वो कौन सी energy है which is needed to roll the stone up the hill. अब एक और तरीका ये भी हो सकता था कि अगर आपको एक huge stone को छोटे stones को break करना है तो you have taken a hammer and broken down the hill. And the other method is that you roll it up the hill और वहां से फिर आप फेंकेंगे तो वो automatically break down हो जाएगा into small pieces. So what is the easier option? Yes, Anya. Is it easier to take a hammer and break it into small pieces, or it is easier to just roll it up the hill and throw it, and once over the hill, the rest of the reaction occurs?
Miss, rolling it up the hill, miss.
Rolling it up the hill. ठीक है. अब क्या किया मैंने? इसको roll किया up the hill. So did that provide an alternate pathway?
Yes, miss.
Exactly. This is exactly the meaning of this sentence over here. जब आपसे ये पूछा जाएगा, you will have to tell the examiner that yes, the enzymes they allow reactions to follow an alternate metabolic pathway. एक pathway था उस reaction को complete करने का, वो क्या था? It will provide a lot of heat. Because if I want to break down a molecule of glucose inside my body, I can just provide a lot of heat, बहुत ज़्यादा heat, around 100 degrees Celsius, और वो molecule टूट जाएगा. But I can't heat my body to 100 degrees Celsius. It has many other disadvantages as well. So the enzyme will bring about the same effect by following a different metabolic pathway. ठीक है? तो ये है और दूसरा क्वेश्चन यहाँ पे क्या लिखा हुआ है कि what happens to the total energy of the substrate and the total energy of the product when the activation energy is lowered? इसका आंसर ये है कि the total energy content of the substrate and the product does not change. The reaction pathway follows a lower energy course. इसका मतलब ये है. Now this graph which you can see, now you tell me, what is plotted on the x-axis?
Miss, the rate of reaction.
The rate of reaction. What is plotted on the y-axis?
Miss, the energy required.
Energy required. ठीक है. And you can see the energy required 0 joules, 2.5 joules, 5 joules, फिर उसके बाद around 7 joules है या 7.5 है, फिर 10 joules. ठीक है. अच्छा. The reaction with enzyme is in the blue pen and the reaction without enzyme is in the red pen. Can you see कि without enzyme जो है वो activation energy कितनी ज़्यादा है?
Yes, miss.
And with enzyme, the activation energy is lower. So in your MCQs and in your structured paper as well, this kind of graph is asked very frequently, in which you might be asked different kinds of questions. आपसे ये पूछा जाएगा कि label the reaction with enzyme, तो आप label कर लोगे. Label the reaction without enzyme, then you will be asked to label the lowered activation energy without enzyme. फिर आपसे ये भी पूछा जाएगा, what is the overall energy which is released during the reaction? Overall energy का भी आपको नजर आ रहा है. This is the part, the overall energy which is released during the reaction. And also you should remember पीछे जाते हैं, the total energy of the substrate and the total energy of the product. The substrate की energy or product की energy change नहीं होती है. The enzyme is only following a lowered energy course. ठीक है? So this is your, ये जो shaded portion है, this one is your overall energy released during the reaction. So you should be very, very clear कि कौन सी चीज किस चीज को show करनी है, right? Any questions from this graph?
No, miss, this is clear.
Clear? And that's why I've written that this graph is very, very important for preparation of paper 1 questions. Okay? Acha. Let's now move on to how to calculate the rate of an enzyme catalyzed reaction. ये कैसे हम calculate कर सकते हैं rate of any reaction? So rate is the speed with which the reaction is happening, right? Now the speed or the rate with which the reaction is happening depends upon how much product is formed per unit time and or एक और तरीका यूज हो सकता है कि how much substrate is disappearing per unit time, isn't it?
Yes, miss.
ठीक है हर रिएक्शन में एक सबस्ट्रेट होता है और एक प्रोडक्ट होता है फॉर एग्जांपल in this reaction the substrate I've shown you is starch and the product is maltose and the enzyme needed to convert or break down starch to maltose is salivary amylase ठीक है तो starch is broken down by salivary amylase to maltose और ये आपके माउथ में रिएक्शन so the substrate is what
Miss, it's starch.
the enzyme is
It's amylase.
and the product is
It's maltose.
maltose. If you have such a reaction, तो अगर मैं आपसे कहूँ कि इस reaction की आप rate determine करोगे तो दो ही तरीके हो सकते हैं ना you can calculate the amount of product formed per unit time ठीक or you can calculate the rate at which the starch is disappearing per unit time. Is this making sense?
Yes, miss.
है ना
Okay.
आपने एक टाइम फिक्स कर दिया जैसे मैंने नीचे लिखा हुआ है 10 मिनट्स ठीक है अब आपके ऊपर है यू कैन डिसाइड लेट मी काउंट और लेट मी जस्ट सी द अमाउंट ऑफ maltose being produced over a time span of 10 minutes or let me judge the amount of starch disappearing over the period of 10 minutes तो दोनों चीजें आपको रेट बता ठीक है ओके go through this
Dependent on the concentration of the substrate. So when the reaction starts, there's a lot of substrate present, which needs to be converted into the product. So all of the active sites are occupied, but then gradually the concentration of substrate decreases. So there are less active sites which are forming the enzyme substrate complexes because a lot of product has already formed. And then when the concentration completely decreases, the reaction stops.
Okay, fine. Did you understand this first point that the rate of any reaction can be measured by the amount of substrate changed or the product formed per unit time? ये समझ आ रही है बात ये दो चीजें
Yes.
This is very, very important because you can be asked to even design an experiment. कोई सा भी आपको example दिया जाएगा या फिर आपको ये you'll be given a choice to choose a reaction and then decide how are you going to measure the rate of the reaction. कोई सा भी आपको खुद सोचना पड़ेगा, right? And that is a very tricky question. हम करेंगे इंशाल्लाह वो. This is the reaction which is in front of you. You can see that a compound whose name is hydrogen peroxide, which is formed normally हमारी body में ये बनता है and it's a very toxic compound, H2O2. This is formed, right? Since it's toxic, the body wants to get rid of it. And we have lots of the enzyme catalase found in the mammalian liver. हमारे liver में ये enzyme है catalase, which can very rapidly, बहुत इसका fast reaction होता है rate, इसका होता है around it can break down millions of molecules of H2O2 very rapidly into water and oxygen. So H2O2 poisonous होता है and water and oxygen are very, harmless compounds. So this reaction happens inside our body, right? The graph is showing you time plotted on the x-axis and the total volume of oxygen collected in cubic centimeters on the y-axis. मतलब कि जैसे जैसे time pass हो रहा है तो कितना oxygen collect हो रहा है. Okay, now you tell me, Alia. इस situation में when we are calculating the rate of reaction, are we calculating the amount of products formed per unit time or we are calculating the amount of substrate disappeared per unit time?
Miss, we are calculating the amount of product formed over time.
Product formed over time. What's the product?
It's oxygen.
Okay. Fine. And you can see that, okay, let me just zoom this picture. Can you just roughly tell me how many cubic centimeters of oxygen is found in the first 30, is formed in the first 30 seconds?
Miss, around 27 centimeter cube.
27,
2.7, Miss,
2.7,
2.7.
2.7,
2.
2.7 centimeter cube. Okay. Acha, after that, what's happening to the rate of a reaction after 30 seconds? Is the rate decreasing or is it increasing?
Miss, the gradient is decreasing, so it means the rate is also decreasing.
Acha, what do you think if I ask you why is the gradient decreasing after 30 seconds?
Because Miss, the concentration of the substrate decreases.
The concentration of the substrate decreases because more and more substrate is being converted into product. So जब substrate ही नहीं रहेगा, so what will the enzymes do? They will be sort of queuing up, line में खड़े हुए होंगे enzymes, ठीक है? और आपके पास कोई फायदा नहीं होगा because all the substrates are converted into products and hence the you can see that the gradient is decreasing और आहिस्ता आहिस्ता जो है वो आपका rate कम होता जाएगा. अच्छा. If I ask you, in fact, यही चीज पीछे भी लिखी हुई है कि the initial rate, initially in the first 30 seconds, it is fast as all the active sites are occupied. ये वो part है 30 seconds का कि जिसमें सारे active sites occupied हैं and the rate of a reaction, अगर मैं आपसे कहूँ कि इस enzyme का rate of the reaction calculate करो, तो I think the most accurate way to calculate the rate of the reaction will be in the first 30 seconds. उसमें सबसे ज्यादा जो है वो आपका रिएक्शन फास्टेस्ट हो रहा है, ठीक है. वरना अगर आप किसी और टाइम स्पैन में 60 सेकंड्स में 150 सेकंड्स में रेट कैलकुलेट करेंगे तो आपको सही नहीं बताएगा, आपको एक सही पिक्चर या एक एक्यूरेट फिगर नहीं देगा अबाउट हाउ फास्ट कैन कैटलेस वर्क. आप समझ आ रही है बात? क्योंकि अगर इफ आई वांट टू नोट, if I want to calculate the performance of catalase, तो performance of catalase मुझे सबसे अच्छी जो है वो first 30 seconds में नजर आएगी because that's the time when we have maximum substrate and maximum active sites available. उसके बाद क्या होने लगता है कि gradient कम होना शुरू हो जाता है because substrate is being converted into product. तो यहाँ से लेकर which means 30 seconds के बाद से लेकर till 390 seconds तक it won't give me an accurate representation of the rate. Is this understandable?
Yes, Miss.
Okay. Acha. Read this also and then I will discuss with you. Catalase, then I have amylase, I have maltase, बहुत सारे enzymes. और सबका हम अलग-अलग calculate कर रहे हैं rate of reaction. And we want to compare how fast, we want to compare the rate of reaction of one enzyme with another enzyme. So the best thing would be that we use the initial rate of reaction as a comparison. Why? Because this is that time period in which the effect of varying factors is not playing a very important role. शुरू में फॉर एग्जाम्पल देखो, शुरू में एंजाइम कॉन्सेंट्रेशन भी आइडियल हो सकती है, सबस्ट्रेट कॉन्सेंट्रेशन भी अच्छी हो सकती है, टेम्परेचर भी सही हो सकता है, पीएच भी सही हो सकता है, बट देन एज द टाइम इज पासिंग द सबस्ट्रेट कॉन्सेंट्रेशन विल रिड्यूस. हो सकता है हमारे पास एक बहुत ही फिक्स कॉन्सेंट्रेशन हो एंजाइम की, तो वो एंजाइम्स खत्म हो सकते हैं, राइट? सो दैट्स व्हाई वी ऑलवेज when we study rates of reaction, we work with initial rates of reaction because this gives us the maximum possible rate of reaction in those conditions. And it also allows us to compare the rate. यानी मुख्तलिफ enzymes के rate अगर हम compare करना चाहेंगे, तो वो भी हमें एक accurate representation देगा because during this time, during let's say in the first 30 seconds, the rate is not affected by the substrate concentration. ठीक है? क्या लिखा है सबसे last sentence में कि if studying the enzyme concentration effect on rate, only the initial rate will give accurate differences in the rates with different enzyme concentrations. इस चीज का हमें पता चलेगा इस ग्राफ से. In fact, you know, the best thing would be that let's go to the textbook and let's read a bit of enzymes from over there. वहां पे हम पढ़ लेते हैं. Yes. This is measuring the rate of formation of a product. यही मैंने आपको अभी ग्राफ दिखाया था, right? Read this first. In fact, you have to read this whole page. I hope you have the textbook, beta. You have the textbook?
Yes, Miss, I have it.
Miss, initially there are a lot of substrate molecules in this solution, so they hit the active site of the enzymes at a very constant and fast rate. Therefore, the reaction is it starts to speed up and gets faster and faster until eventually the number of substrate molecules reduce and less substrate molecules hit the active site. Therefore, the speed of the reaction decreases gradually and eventually stops at 7 seconds.
At
Sorry,
how?
390 seconds.
390 seconds. Okay, fine. Achha, if we read this, it says the reaction begins very quickly. As soon as the enzyme and substrate are mixed, the bubbles of oxygen are released. ये शुरू में बहुत fast होता है, right? And in fact, when we do practical 3, paper 3 with students in school, so this reaction is shown to them. It's very easy to show this reaction because if you take, although this enzyme is found in the mammalian liver, in the human liver, it is also found in the liver of any animal. So you isolate liver, and liver has catalase, and you take just small pieces of liver, kaleji jo hoti hai, and you pour H2O2 on it, and you will suddenly see bubbles of oxygen being released, which shows that the enzyme is there in the liver, and then H2O2 agar hoga, wo react karega, and the bubbles of oxygen are produced. Okay? Of course, we will see that a large volume of oxygen is collected in the first minute of the reaction, and as the reaction continues, however, the rate at which the oxygen is released, it slows down, the reaction gets slower and slower. ये सब चीजें तो हमें इस ग्राफ में नजर आ रही है, but then the examiner, what he will do, he will give you this graph and he'll ask you why is the rate slowing down. ठीक है? Always remember, examiner आपको enzyme वाले question में जब भी graph देगा, he wants two kinds of information from you. Number one, he can ask you to describe the graph. If he is asking you to describe it, you will just quote the data from the graph. यानी कि अगर मुझे कोई कहता है कि इस ग्राफ को डिस्क्राइब करना है, I'll start describing it from 0 seconds. कि I would say कि अच्छा 0 seconds पे rate इतना था, फिर 30 seconds पे the amount of oxygen collected was 2.7 cubic centimeter, फिर 60 seconds पे इतना कम हुआ, फिर 40 पे इतना कम हुआ, and then by the end, I will say by the end of 390 seconds, the slowly and slowly the amount of the oxygen that was collected per unit time was being reduced. लेकिन इसमें कहीं पे भी मैंने reason नहीं बताया है because the examiner is only asking me to describe the graph. If he asks me to explain the graph, this is what I will say. ठीक है? एक चीज याद रखनी है, and both of you can note it down, that whenever I'm just writing it down over here. ठीक है? For explaining a graph, you have to keep these points in mind. Explaining a graph, sorry, let me rewrite it. For explaining a graph on enzymes, we have to keep the following key points in mind. Right? Acha. Whenever any student is given a task to explain a graph on enzymes, the answer will always be incomplete if the key points, the key words, not even the key points, the key words that I have written in my blue pen right now are not mentioned. ठीक है? कैसे? Now look, I'm reading the paragraph and you will see how the usage of these keywords is important while explaining the cause of an enzyme catalyzed reaction. ठीक है? Step number 1. सबसे पहले, this is step 1 and I am writing down number 1 over here. When an enzyme and substrate are mixed, there are a large number of substrate molecules. शुरू में, within the first 30 seconds, जब आप enzyme substrate को mix करते हो, so you have a large number of substrate molecules. At any moment, almost every enzyme has a substrate molecule in its active site. The rate at which the reaction occurs depends upon two factors. What are those? कितने हैं हमारे पास enzyme molecules in the beginning, and the speed with which the enzyme can convert the substrate into the product, release it, and then bind to another substrate molecule. So the reaction begins. In the first 30 seconds, it is very, very fast. Why? Because at any point in time, whether it's the first 30 seconds or any other 30 seconds or 40 seconds, the rate of an enzyme catalyzed reaction depends upon the concentration of the enzyme molecules and the speed at which the enzyme can bind to a substrate, convert it into a product, release it, and then bind to another substrate molecule. ठीक है? लेकिन, however, as time is passing and more substrate molecules are converted into product, there are fewer and fewer substrate molecules to bind with the enzyme. यानी कि जाहिर है enzyme जब तेजी से सब reaction कर रहा है, तो it is gradually finishing off all the substrate molecules. क्योंकि enzyme तो recycle हो सकता है, लेकिन substrate तो नहीं recycle हो सकता है. We cannot have an infinite concentration of substrate in any experimental setting. ये इसका जवाब ये है कि किसी भी experimental setting में आपके पास एक indefinite या infinite concentration substrate की नहीं हो सकती है. इसलिए there will be a time when your substrate molecules are finished. Now when they are finished, अब examiner wants कि आप इसको ये बताएं examiner को कि enzyme molecules will be waiting, or in other words waiting in a queue, एक line में खड़े हुए हैं enzyme molecules, so that they can hit their active sites. And as fewer and fewer substrate molecules are left, the reaction gets slower and slower until it eventually stops. G, Alia and Amna, is this thing understandable जो मैंने आपको अभी समझाया है?
Yes, miss.
Okay, I want both of you, Alia also and Amna, to explain this entire thing to me separately. दोनों अलग-अलग से बताओ ताकि ये आपको अच्छी तरह से आ जाए. Yes, Alia, your turn.
So, miss, I just explain all these terms which are given here.
Everything which I've explained, जो मैंने यहाँ से लेकर, when I was explaining you this paragraph from here till finally the reaction stops. Yes.
Okay, miss. So basically, miss, the rate of reaction depends on the number of enzyme molecules which are there in a solution, and then the speed of the enzyme. So initially, when the substrate molecules are added, the concentration of the substance is very high, which is why the rate of reaction is initially also very high, because all the active sites of the enzymes are occupied. But then as the concentration of the substrate decreases, because they are being converted into products, uh, the enzyme activity also decreases, so the rate of reaction will be lower. And then when there are no substrate molecules left, the reaction completely stops because everything has been converted into the product.
Correct. Yes, Alia. Sorry, Amna.
Miss, first, when the reaction starts initially, there are a lot of substrate molecules, and since there are a lot of substrate molecules, meaning there are more than the enzymes present, the enzymes have their active sites full of the substrate, and as they're gradually turned into products, the number of substrate, concentration of substrate decreases because they're not present in an infinite amount. The speed of reaction decreases and eventually stops. The limiting factor of the reaction is the number of molecules present.
Molecules
Enzyme
of what?
molecules present.
No.
Molecules.
Wait. Is the limiting factor the number of enzyme molecules or the substrate molecules?
The limiting factor is the substrate molecule.
Exactly, the substrate molecule. You have to be very, very careful. जब आप limiting factor की बात लेके आते हैं ना, you have to be very careful. Look here. as the reaction is proceeding, the rate is slowing down. आपसे पूछा जाएगा क्यों? तो आप कहते हैं कि the substrate is converted into products. Then who's the limiting factor? Substrate. Because अगर इस वक्त मैं थोड़ा सा और substrate डाल दूं इस situation में, the reaction will change. थोड़ा सा reaction और तेज हो जाएगा, ऐसे हो जाएगा. If I put more substrate, if I put even more substrate, maybe the reaction goes like this or like this. ठीक है? तो क्या चीज limit कर रही है? What is that factor? कि जो लिमिट कर रही है उसके रेट को आफ्टर दिस पॉइंट में बी इज द concentration of the substrate. Enzymes तो हैं, but what did we say over here? We said the enzymes are waiting for the substrate molecules to hit their active sites. ठीक है? Are you understanding this, Amna?
Yes, yes.
ठीक है? अच्छा, and then this is okay. So Acha. Now the question is, the question in your textbook is why is it better to calculate the initial rate of a reaction from such a curve as the one in figure 3.6 than simply by measuring how much oxygen is given off in the first 30 seconds? Yes. Amna, would you like to answer this?
This maybe because that way we can determine how fast it actually is. Because if we do that based on the amount of oxygen collected, it would be inaccurate because some oxygen can escape into the
Exactly.
atmosphere.
And also another thing in that, why are we only choosing the initial rate and not the rate at any other time, is because the during this time period, during the first 30 seconds, the reaction is the fastest, it's the steepest, so it will give us a more accurate reading. And during this time period, the effect of various factors, जैसे कि effect of pH हो गया, effect of temperature, effect of substrate concentration, ये इतना नहीं मैटर करता जितना कि 30 seconds के बाद मैटर करेगा, क्योंकि 30 seconds के बाद the picture will be different, right? Acha, now is the concept of how much time is left. Okay, ठीक है, हम ये कर सकते हैं आराम से. We still have 10 minutes. So, did you guys study about a colorimeter in O levels? What a colorimeter is? Yes or no?
Yes, I didn't.
Acha, what about you, Alia?
Or not really, miss.
अच्छा ठीक है, तो हम कर लेते हैं इसको, because this is something which is new to the syllabus. ठीक है, सबसे पहले हम देखते हैं what is a colorimeter. So it's an instrument whose job is to measure the color of a solution by measuring the absorption of different wavelengths of light. ठीक है, अब इसमें बहुत सारी चीजें हैं जो हो सकता है आपको ना आती हो. What do you understand by the wavelength of light? Yes, Alia, what is the wavelength? Physics में किया होगा आपने.
Miss, it's basically the distance between 2 points in the light wave, two adjacent points.
Okay. What's the wavelength? The distance between these 2 points or these 2 points? Yes. ठीक है, that's the wavelength. ठीक है. For example, कि ये है आपका एक bulb. Okay. This is your bulb and light is coming out of it. The bulb has its certain energy, which is watts. है या नहीं है? Watts होते हैं ना, energy of the bulb, power of the bulb in fact. So let's say this is a 60 watt ka bulb and it is emitting light energy. Light energy दे रहा है. So you have a 60 watt bulb. Let me do like this. This is a 60 watt bulb. This is a 100 watt bulb. Which will give energy of more light intensity? Yes, girls?
Miss, the 100 watt bulb.
100 watt, right? So light के over here, अभी तक के लिए मैं आपको समझा रही हूँ कि there are many properties of a light, but right now I am just bothered about two properties. And what are those properties? The intensity of the light, जो कि एक bulb से निकलती है, and the other is the wavelength of the light. ठीक है? And wavelength you define. और wavelength जो है, of course, लाइट की वेवलेंथ ऐसे भी हो सकती है, the wavelength can be different also like this. Okay, so it depends upon के वो लाइट कौन से मीडियम से पास हो रही है, राइट? लाइट कैन पास थ्रू सॉलिड्स, थ्रू लिक्विड्स, through gases, and light can also pass through different colors. For example के हमारी जो visible light है, अब sunlight की बात करते हैं हम, this is the sunlight, and in physics you must have done the electromagnetic spectrum. Did you do this in O levels?
Miss, we
Yes.
just touched the topic.
You just touched the topic. So इस electromagnetic spectrum में ना we have various electromagnetic rays जिसके बीच में कहीं ना कहीं light rays fall करती हैं. कौन सी वाली light rays? Visible light, जो हमें जिससे हम देखते हैं इस दुनिया में, right? The white light is a visible light, and you all know that that white light is made up of 7 colors. Yes. ठीक है, ये हो गया आपके 7 colors. Do you guys know the abbreviation of this? Vibgyor, violet, indigo, blue, green, yellow, orange, red. Do you know this?
Yes, Miss.
Right? So sunlight, which is apparently looks like white light, is made up of actually those 7 colors, because it can split into those 7 colors. और इस हर color की एक wavelength है, right? अगर हम उसकी पूरी range ले लेना, for example, if I start from this end and ये वाला end जो है वो violet है, उसको मैंने blue pen से किया है, and this end is red. ठीक है? So the light wavelength ranges from, जहाँ तक मुझे याद है, if I'm not mistaken, it's from 400 nanometers to 700 nanometers. ठीक है, ये visible light की है. And I think I've done this topic with you, Amna, when we were doing electron microscopy, remember?
Yes, Miss.
Right? So this is the range of the wavelength of visible light. और ये range depend करती है कि कौन से color के through ये light waves pass होती है, right? अच्छा, so this was just the background that I have given you about 2 properties of light. One is intensity and the other is wavelength. याद रखें आप कि दोनों separate चीजें हैं. The wavelength is different and the intensity is different. Intensity depends upon the power of the bulb. ये कितनी intensity का bulb है. Wavelength depends upon through which color that light is passing. ठीक है, हर color की अपनी एक अलग wavelength है. अच्छा, going back to where we were, why are we doing colorimeter? क्योंकि ये एक instrument है या एक device है who is measuring the color of a solution. Color of a solution को measure कर रहा है by measuring the absorption of different wavelengths of light. For example, कि I am doing Benedict's test. You all know what Benedict's test is, or do I need to tell you what that is?
So we did that.
We did that because when we were doing biological molecules, so we did that. For example, we are doing Benedict's test और उसमें हमारे पास एक range of color आती है. ये red है, and let me just choose another color. We get light blue and we get orange. ठीक है, orange आता है. Now these are separate colors. ठीक है, I can measure the rate of a reaction by colorimeter. कैसे? कि I will measure the color of a solution by measuring the absorption of different wavelengths of light. This solution will measure light, will allow the light to pass through it in a different manner क्योंकि इसका color blue है. इस solution का color orange है तो इससे light जो है वो different तरीके से absorb होगी या different wavelength की light absorb होगी और इसके through different wavelength की light absorb होगी. So by measuring the absorption of different wavelengths of light, I can measure the rate of a reaction. कैसे? और especially वो rate of reactions मैं measure कर सकती हूँ जिसमें के हमें change of color नजर आ रहा होता है, right? So this means that a colorimeter is an instrument that allows us to measure the rate of a reaction, especially those reactions which involve a change of the color of a solution over a period of time. How? By measuring the absorption of different wavelengths of light. किस तरह से we are going to see this quantitatively. ताकि पीछे हम कोई miss कर लिया है point. हाँ, sorry. It says, if the method, I'm reading from over here, if the method used for measuring the progress of an enzyme controlled reaction involves a color change यानी कोई भी हम जैसे कोई एक रिएक्शन कर रहे हैं जिसमें वो रिएक्शन हमें नजर आ रहा है कि इसमें कोई कलर चेंज हो रहा है, ठीक है, देन द कलरीमीटर कैन बी यूज। अगर कोई ऐसा रिएक्शन है जिसमें वो रिएक्शन जो है ना वो कलरलेस है, तो फिर कलरीमीटर का कोई यूज नहीं है, राइट। द नेम ऑफ दिस इंस्ट्रूमेंट इज गिविंग यू एन आइडिया कि ये सिर्फ उस वक्त आपको काम आ सकता है व्हेन देयर इज एन ऑब्जर्वेबल कलर चेंज, ठीक है। उस वक्त यू कैन यूज दिस कलरीमीटर टू मेजर द कलर चेंज क्वांटिटेटिवली। हाउ? This will give you numbers that can be plotted on a graph. A colorimeter is an instrument that measures the color of a solution by measuring the absorption of different wavelengths of light. The greater the absorption of the light, the greater the concentration of the substance causing the color. ये समझ आ रही है बात? जाहिर है जितना thick color है इसका मतलब उस substance की concentration ज्यादा है और उतनी ही ज्यादा जो है वो light absorb करेगा. And this can be, done quantitatively. कैसे? Look here. Right now, in the beginning, I gave you the example of starch. जब Amna, when you were not there, I was discussing with Alia. ये एक बिल्कुल standard सा reaction है जिसमें हमारे पास starch है, and it is broken down by salivary amylase into maltose. ठीक है? अब जब starch maltose में change हो जाएगा, तो of course maltose is not going to give you a positive test for iodine. In fact, maltose आपको एक positive test देगा Benedict's reagent के लिए, because maltose is a reducing sugar. ठीक है? So what does it say in the amylase starch experiment described in practical activity 3.1? You can measure the intensity of the blue-black color obtained in the iodine test using a colorimeter. The color acts as a measure of the amount of starch still remaining. यानी कि मेरे पास एक dish में starch है या एक test tube में starch है, and I put some amylase into it, और अब रिएक्शन स्टार्ट हो रहा है. जाहिर है इनिशियली तो फौरन पूरा का पूरा स्टार्च कन्वर्ट नहीं होगा, राइट, एंड इट विल टेक टाइम फॉर द स्टार्च टू बी कन्वर्टेड completely into maltose. So over the period of time, I can measure the intensity of the starch present by allowing that starch solution to be passed through a colorimeter, और जितना डार्क उसका कलर होगा उतनी लाइट वो एब्जॉर्ब करेगा, ठीक है, और जैसे जैसे टाइम गुजरेगा उसकी एब्जॉर्प्शन ऑफ लाइट कम होती जाएगी. And that degree of absorption of the light is actually, it can be read off from a digital display that is shown on a colorimeter. और वो digital display जो है वो हमें बताएगा कि कितनी light absorb हो रही है over the period of time. Is this understandable to you girls?
Yes, miss.
What about you, Alia?
Yes, miss.
Alia, do you have a class today?
No, miss, I don't have a class today.
Acha, so we can at least finish this topic. See, it's showing you a light. Then the light is made to pass through a filter, कोई भी colored filter जैसे red light, and then the solution in a tube or cuvette, and then the light meter, and the readout is 0.56. ठीक है, अब आपको ये दिखाया है इन्होंने starch वाला, ठीक है. अच्छा, you have 5 test tubes. If we start from the test tube on the left hand side, test tube number 1 still is black in color. इसका मतलब starch has not been broken down to maltose. Still black, still black. It's becoming a little bit on the browner side, and now the last test tube shows कि ये जो है ना आपका starch completely digest हो चुका है into maltose. So we can allow the light, we can take these test tubes. इस test tube को हम क्या कर सकते हैं, इस setup में रख सकते हैं, ठीक है. For example, मैंने इसी test tube को इस setup में रखा, it gave me this reading, ठीक है. फिर थोड़ी देर बाद it will give me another reading, and then another reading, and another reading, and then finally another reading. Every time the absorption of the light will keep on decreasing. जैसे अगर 0.56 आई है तो 0.52 आएगी, 0.5 आएगी, और आहिस्ता आहिस्ता इसमें लाइट इतना नहीं एब्जॉर्ब होगी. एंड देन वी कैन हैव अ क्वांटिटेटिव मेजरमेंट. इससे फायदा ये होगा कि हमारे पास एक क्वांटिटेटिव वैल्यू आएगी which can show me the progress of an enzyme catalyzed reaction. So a colorimeter is a useful instrument which can enable me to quantitatively monitor the course of an enzyme catalyzed reaction, especially उन tests में जिसमें के color show हो रहा होता है, right? If you do this over a period of time, you can plot a curve of the amount of starch remaining against time. You can then calculate the initial reaction in the same way as you did for catalase and hydrogen peroxide. It is even easier to observe the course of this reaction if you mix starch, iodine solution, and amylase in a tube and take regular readings of the color of the mixture in this one tube in a colorimeter. However, this is not ideal because iodine एक ऐसा chemical है which interferes with the rate of the reaction and slows it down. Because when we put iodine to test for the presence of starch, the iodine actually slows the progress of a reaction because iodine is a toxic substance. ठीक है. Did you understand this, girls?
Yes.
Yes, miss.
Are you sure, both of you, there's no confusion?
No, miss.
No? Acha, okay.